Transfer methods in Southern blotting. There are different types of the transfer methods of DNA to the membrane. The most common transfer methods, they are upward capillary transfer, downward capillary transfer, simultaneous transfer to two membranes, electrophoretic transfer and vacuum transfer. So these are some of the common transfer methods by which DNA is transferred from gel to the membranes. One of the most common method of transfer of DNA from gel to the membrane is upward transfer. Here we can see that how upward transfer is performed. In this diagram, this is the tray in which there is the transfer buffer. On the tray, there is the gel. Here we can see that this is gel. On the gel, there is the membrane. This is the membrane. We want to transfer DNA from this gel to this membrane. On the membrane, there are Whitman tissue papers. Here we can see. Then there are additional paper towels. On the paper towel, there is glass plate, and on the glass plate, there is weight. So what happened in the tray? Here there is a buffer, and this buffer will move upward, and the DNA bands which are present on the gel, they will bind, they will transfer on the membrane. So this type of the transfer of DNA is called as upward transfer, because the DNA bands are present in the gel. These bands, they will transfer to the membrane. Here we can see upward transfer of DNA to membrane. The buffer will move upward. We can see here with arrows, with red arrows, and with blue arrows we can see the bands from the gel will be converted to the membrane. So this type of the transfer of DNA from gel to the membrane is called as upward transfer. So this is also one of the common method which is used for transfer of bands from gel on the membrane in case of Southern blotting. So if we conclude that there are different methods by which we can transfer DNA bands from the gel to the membrane, most common methods, they are called as upward capillary transfer, downward capillary transfer, simultaneous transfer on two membranes, electrophoretic transfer, as well as vacuum transfer.